So now bacteria are being used to solve murders, apparently. We're going to talk all about this today on today's episode of Short Stories of Bacteria. Hey everybody, welcome back to another episode of Short Stories of Bacteria. I, as always, am your host, Dr. K, and I'm here to guide you through yet another story chronicling the lives of the tiny and the infamous. That would be, of course, the amazing and fascinating bacteria. I have such a cool story to share with you guys today. It's super neat. It came out of Arizona State University. It's a groundbreaking new paper, and I'm so excited to share it with you. It's talking about bacteria and murder, so that's a really neat combination. But before we jump into that, don't forget to hit that like, share, subscribe button. If you're listening on Spotify or if you're listening on Apple Podcasts, don't forget to hit that follow button. Super, super useful for the channel. So thank you very, very much. But enough of that. Let's jump into the science for today. Really, really cool stuff. Like I said, I'm really, really excited to share this with you guys. Before we actually talk about the paper, though, I want to emphasize this point because it's a broader cultural point that I've noticed before, and it's just interesting. And that is that our culture, and I think it's been going on for a very long time, obviously at least since Sherlock Holmes showed up, right? Our culture is extremely obsessed with crime, right? What are the types of psychological things that drive someone to commit a crime, right? What are the types of environmental factors that drive someone to commit crime? What types of crimes are there? How do people commit the crimes? How do people go about getting away with crime? There's literally a TV show called How to Get Away with Murder or something like this, right? On the other side, how do you go about capturing a criminal? How do you go about identifying who the criminal was? How do you go about putting someone in jail, right? If you turn on the TV at any point in time during the day, I'm sure at some point during your scrolling, you'll run into some kind of procedural crime drama. How do you go about locating the criminal, identifying the criminal, capturing the criminal, and then convicting the criminal, right? So in addition to all that, I know we spend a lot of time thinking about the psychology of crime, but there's a lot of emphasis on the science of crime too. I mean, obviously, I'm sure in a lot of TV shows you'll see, oh, look, it's the forensic scientist person. They're like getting DNA sequenced in like 5 seconds, which never actually happens. But the main point being is that forensic science, that's totally a real thing, and it's really, really cool. And this is my first point, a lot of time, a lot of money goes into funding research and learning more about not only the psychology of crime, but the science of crime. How do you go about identifying a criminal? And in particular, we talk about this within the context of murder, because murder is like one of the most egregious crimes that we have. And so, yeah, and this in particular is related to bacteria, as we'll see in a second. Now, honing in on murder a little bit, obviously, I mean, the one thing that I hear over and over again on all these procedural crime dramas is you need means, motive, and opportunity, right? And in particular, I want to hone in on opportunity, right? Because one of the most important things when you're understanding a murder and who gets murdered is who got murdered, who is doing the murdering. Jeez Louise, I'm all over my toes today. Is establishing when that person died, right? So let's say you have a person and they get murdered, right? It's really important for you to know when that person got murdered, right? So you can know who had the opportunity to kill that person, right? Obviously, like if you don't know when the person died, then you have no way of knowing who had the opportunity to kill them, right? So that would be any person during the course of the day. So time of death is really, really important. Now, a lot of science actually goes into understanding when the time of death occurred. You guys probably heard of rigor mortis. Actually, it's actually kind of cool. So the way that rigor mortis works is your body physically runs out of ATP, the energy currency of the cell. And so then as they run out of ATP, they exist in this contracted state. So it's this really stiff, held together state, right? Because all your muscles are just held in this contracted state, right? And so they stay there. Right. And so then that's what rigor mortis is. It's you've lost all your energy, and so you stiffen up. Now, that's one aspect of it. Another aspect of predicting time of death has to do with decomposition. How much has a body decomposed, right? And you can maybe try and back calculate, say, okay, it's, 10% decomposed. So based off of that, then we can make a prediction about when that person died. But there are some limitations when you're trying to figure out the time of death based off of decomposition, right? Was the body transported, right? Was it in really, really cold conditions for a while? That influences the rate of decomposition. Was it in really obviously warm environment for a while? That could change the rate of decomposition, right? And so there's some natural limitations that occur when we're trying to establish the time of death based off of the decomposition process. So point number 2, I guess, real quick, just to emphasize this, time of death, especially within the context of murder, right? Time of death is super, super important. And decomposition is a great way of identifying the time of death. Okay, even if there are some limitations on it. And since there are some limitations still left on the time of death, an emergent question that shows up whenever you're doing forensic sciences is, are there some better or are there some more consistent ways of identifying the rate of decomposition? Are there some better, more consistent ways of identifying the rate of decomposition? And this is where we're going to bring in Arizona State University. Now, like I mentioned, this is a groundbreaking, brand new paper, super, super cool stuff. And what they were doing is they were trying to address this question. Are there better, consistent ways that you can identify the rate of decomposition? And they approached it through the lens of bacteria, right? Now, obviously, in order to understand this, we need to talk a little bit about the whole decomposition process. As it turns out, everything that is alive is going to die at some point, right? And when those live things die, they will start to be decomposed. And the things that are doing the decomposition, those are bacteria. They tend to be bacteria, right? So point number 3, all things die. All things decompose. And that decomposition tends to happen through bacteria. Now, if you were an enterprising young scientist, or mayhaps even an enterprising older scientist, whichever the case may be, you could be thinking to yourself, well, gee, is there any way I could relate this decomposition process, and in particular the bacteria associated with the decomposition process? Is there a way that I could relate that to this time of death thing that we're studying? Because you can imagine it'd be really, really useful. It'd be really, really useful if there was a certain type of bacteria, right, that showed up, right, whenever someone died, right at the start of decomposition, right? Let me see if I can paint a picture for you. So let's say that poor Joe Bob dies, right? And then immediately after he dies, a bacteria colonizes and starts the decomposition process, right? Then, hold that thought, then let's say that as he starts the decomposition process, by starting that process, you're establishing a new ecological niche for a second type of bacteria to show up. And then together, they start establishing a new ecological niche that allows a third type of bacteria to show up. And what I want you to envision here is that over the course of the decomposition process, especially starting from the death of the individual, right, it'd be really useful if you could make predictions, or if you could use bacterial presence, if you could use bacterial richness as a way of predicting when the time of death was. Okay, so that's point number 4. I'm going to say that again. It'd be really, really nice to use bacterial presence and bacterial richness as a way of identifying when the body started decomposing. Okay, and that's exactly what the folks over at Arizona State University did. Good job, folks over at Arizona State University. What they did is they started doing DNA sequencing over the course of a decomposition process, looking at the bacteria that were present at each of these stages of the decomposition process. And what they found was absolutely wild. They found that there are certain types of bacteria, right? And not only just types of bacteria, these are rare species of bacteria, right, that are showing up at the initial phase of decomposition, right? So like you can imagine a situation where it's like, okay, all right, there's a bacteria, it's at the initial start of decomposition, but it's found all over the place, so that's not really helpful to us, right? Or there's a really rare bacteria, but it doesn't really hang out with a person after they die, so that's not really. What we have here is we have a really rare bacteria that doesn't show up all that often, but it does show up right when the decomposition process starts. Right. In fact, there's a whole series of different bacteria that grow together right at the start of decomposition. Right. So that's really, really useful. Now, what's even cooler, well, not even cooler, but it's related to this, is that these rare bacteria, not only were they found in the case of human decomposition, but they were found in a whole bunch of other animals as well. Things like, what I have here? So I have pigs, rats, a bunch of other mammals as well. So this hints at the idea that there could be a series of rare bacteria, right, that are universal decomposers, right, that they start the decomposition process. Now, relating this back to forensic science, what the researchers then did is they took the bacteria that were present and then took the richness of the bacteria that were present, right? And then they used a machine learning algorithm, which goes way beyond my pay grade, and I don't know the math behind that. But they plugged it into this machine learning algorithm, and they used this algorithm in conjunction with the species and the richness of those species as a way of making these predictions as to when those bodies actually died initially. Right? So point number 5, really, really cool. Based off of the growth rate of these specific and unique bacterial species, the researchers could figure out when a body died. It's such a cool paper. It's a really neat instance of how, first of all, it's a really neat instance of how bacteria are going to be staying with you even after you die, and that's totally fine. They got to do some decomposing. But it's really neat that we can use those bacteria, these rare bacteria, and how far along they've grown and the types of bacteria that are present as a way of establishing the time of death. So let's wrap this up. Point number 1, a lot of time and money goes into researching the psychology and the forensic science of crime. Point number 2, time of death is extremely important when we're talking about murder, obviously. But there's some limitations on how we go about establishing time of death. Point number 3, everything dies and everything gets decomposed, and that decomposition is mediated by bacteria. Point number 4, it'd be really, really awesome if we could use bacterial presence and richness of bacteria as a way of establishing when a body started decomposing and thus when a body died. And finally, point number 5, based off of the growth rate of specific and unique bacterial species, these researchers were able to essentially establish the time of death of these mammals. So really, really cool story, really neat instance again of how we can use bacteria as a way of establishing time of death and how these really tiny bacteria are being used as a way of, or being useful in establishing who the criminal was in a murder. So, hey, next time you're doing like a murder mystery or something related to that, just try bringing bacteria into the conversation. Maybe you'll catch the killer. But that is enough nonsense out of me. Thank you guys ever so much for tuning in for another episode. And I'll see you guys again next week on another episode of Short Stories of Bacteria. Hey everyone, thanks for watching that video. If you guys enjoyed it, if you liked what you saw, don't forget to hit that like, share, subscribe button, and I will hopefully see you guys in the next video. Later!